Pinch the skin on the back of your hand. Pull it up about a centimeter. Hold it for 5 seconds. Now let go, and count how long it takes to lie flat again. That is a clock. A crude one, but a clock. The slower the skin settles, the more divisions the cells underneath have completed, and the more bookkeeping has piled up inside them. I spent most of my life looking at the small things. Quarks, photons, the path integral. I never quite got around to looking at the small things inside my own body until the body itself stopped working. So let me tell you what is going on in there. Right now, in every cell, you have something like 30 trillion of them. Not all are dividing. The neurons in your brain mostly do not. The cells in your heart mostly do not. But the rest, the cells in your skin, your gut lining, your bone marrow, your blood vessels, your immune system, are dividing constantly. Every minute of every day, somewhere in your body, 2 cells are becoming 1 cell, becoming 2 cells. And every one of those divisions leaves a mark. Picture a single cell, not the whole thing, just the nucleus. A spherical compartment about 6 micrometers across, holding 2 meters of DNA folded into 46 chromosomes. At each end of each chromosome, there is a cap. Think of the plastic tip on the end of a shoelace. The cap is a short sequence repeated thousands of times. TTAGGG, 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 over and over. About 10,000 base pairs of this repeat sit on the end of each chromosome when you are born. That cap has a name, the telomere. Now look closer. Run your eye down the strand, past the telomere, into the working part of the chromosome, the part that codes for proteins, the part that runs you. Scattered along the DNA, like sticky notes attached to a long manuscript, are small chemical marks. Methyl groups, a carbon atom with 3 hydrogens bolted onto a cytosine base. Each one tells the cell whether that particular gene is on or off, loud or quiet, available or hidden. Step back further, outside the nucleus, in the cytoplasm. Floating in there are several hundred small football-shaped bodies, the mitochondria. Each one has its own loop of DNA, its own membranes, its own machinery for taking sugar and oxygen and turning them into the molecule that powers everything you do. Walking, thinking, pinching the back of your hand. And then, throughout the tissue all around this one cell, there are other cells, some young, some old, and some that have stopped. They have stopped dividing, but they have not died. They sit there, quiet on the outside, screaming on the inside. These are the 4 clocks. The telomere clock, the methylation clock, the senescence clock, the energy clock. Each one ticking at a slightly different rate, in a slightly different language. Each one telling a part of the story of how long you have left. The funny thing is, none of these was being measured when I died. We only knew about the first one in any detail, and even that one we had only begun to understand. The rest came after. So let me show you each one in turn. Slowly. Because the order matters, start with the telomere, that cap on the end of the chromosome. It exists because of a problem in copying DNA. When a cell divides, it has to copy its entire genome. The machinery that copies DNA cannot quite finish the job at the very end of each strand. It runs out of footing. A few dozen base pairs at the tip get left uncopied. So if you started with a strand and just copied it over and over, the strand would get shorter every time, and eventually it would eat into the genes themselves. Evolution solved this with a buffer. The buffer is the telomere. 10,000 base pairs of meaningless repeat at the end. You can lose 100 of them and lose nothing important. You can lose 1,000 and still be fine, but not forever. In 1961, a young researcher named Leonard Hayflick, working at the Wistar Institute in Philadelphia, was growing human cells in dishes. Everyone before him had assumed that if you fed cells properly and kept them clean, they would divide forever. They did not. Hayflick watched them. He counted. Around 50 divisions, give or take, the cells stopped. They did not die. They did not get sick. They just refused to divide again. He published this, and most of the field thought he had made some mistake with his technique. He had not. He had discovered that cells count. What were they counting with? It took another 20 years to find out. Elizabeth Blackburn, then a young biologist at the University of California in San Francisco, was studying a strange little single-celled organism called tetrahymena. It had many small chromosomes, and Blackburn worked out their endings. She found that repeat, TTAGGG, over and over. Then her graduate student, Carol Greider, on Christmas Day of 1984, ran the experiment that revealed the enzyme that builds the repeat. She mixed a cell extract with a synthetic primer and found that the primer was being extended. Something in that broth was adding telomere sequence onto the end of a DNA strand. They called the enzyme telomerase. Blackburn, Greider, and Jack Szostak eventually won the Nobel Prize for it. That was in 2009, long after I was gone. Here is what we know now. Every time one of your dividing cells finishes a division, the telomere at the end of each chromosome shortens by something between 50 and 100 base pairs. Starting at 10,000 base pairs at birth, a typical cell can divide maybe 50 or 60 times before the telomere gets short enough to trigger an alarm. When that alarm fires, the cell stops dividing. It enters what is called replicative senescence. It cannot reproduce, but it does not die. Most cells in your body do not have telomerase. They cannot rebuild the cap. So they ration their divisions. Some cells do have it. The germ cells in your reproductive organs, the stem cells in your bone marrow and your gut lining, and cancer cells, almost always. That last one is the reason your body does not just turn on telomerase everywhere. The cap is a safety device. If your cells could divide forever, the ones that wanted to keep dividing, the tumors, would have nothing to stop them. Researchers can measure telomere length now from a small blood sample. They take your white blood cells, extract the DNA, and use a technique that gives an average length across all your chromosomes. The number comes back in kilobases. A young person, 8 or 9. A middle-aged person, 6 or 7. A person past 80, often 4 or 5. There is a great deal of variation between individuals. Some people of 70 have the telomeres of a 40-year-old. Some 40-year-olds have the telomeres of a 70-year-old. That variation is the part that interests the people studying lifespan. Now, while the chromosome has been shortening, something else has been happening along its length. Quiet. Steady. The methyl groups have been moving. I told you about those methyl groups. The small chemical sticky notes attached to certain cytosine bases. There are roughly 28 million sites along your DNA where a methyl group can sit. Whether one sits there or does not is what tells the cell whether to read that stretch of DNA as a gene or to ignore it. Methylation is how a liver cell knows it is a liver cell and not a brain cell, even though both have the same DNA. The methylation pattern is the difference between the two. The pattern is not static. It shifts slowly over the course of your life. Some sites that were methylated when you were a child lose their methyl groups. Some sites that were unmethylated when you were a child gain them. The drift is small at any given moment, but it adds up. And the pattern of the drift is extraordinarily consistent from person to person. In 2013, a biostatistician named Steve Horvath at the University of California in Los Angeles did something very simple and very powerful. He took methylation data from thousands of people of known age across many tissues. He selected 353 specific methylation sites scattered across the genome that drifted in a predictable way with age. He fit a mathematical model to them. Then he tested it on new samples. The model predicted a person's chronological age, from a blood draw or a saliva sample, to within about 3 or 4 years. This was the first epigenetic clock, There are now several. Morgan Levine, who worked with Horvath and is now at a longevity research company called Altos Labs, built two more. One she called PhenoAge. Another she called Grimage. These ones do something different. They do not just predict your chronological age. They predict your biological age, which is to say, how far along the road to your own death you actually are. Grimage was trained against mortality records, against measured plasma proteins like adrenomagellan and growth differentiation factor 15, and against smoking pack years. Two people of 50 can have very different Grimage scores. The one with a higher score has, on average, less life remaining. In some studies, Grimage predicts all-cause mortality in the next decade better than blood pressure, better than fasting glucose, better than self-reported smoking status. Think about what this means. The cell is keeping a record. Every cigarette you smoked, every year of bad sleep, every chronic infection, every decade of low exercise, every drink of alcohol, every season of unmanaged stress, these things change the methylation pattern in a measurable way. The clock can read the changes. The clock is mostly accurate. People who score old for their chronological age really do, on average, die younger. People who score young really do, on average, live longer. The clock can also move backwards. This is the part that took the field by surprise. If a person of 50 starts exercising 5 days a week, sleeping 8 hours, eating mostly plants, and managing their stress, their methylation pattern shifts, and a year later their Grimage has moved down. Not by a lot. Maybe a year, maybe 2. But it moves. The drift is not one way. The clock is responsive. It is not measuring something that has already happened and cannot be undone. It is measuring something that is being continuously written and rewritten every day by what you do. The methylation pattern is one piece of bookkeeping. The senescent cells are another. And the two are not separate stories. They are pages of the same book. Go back to Hayflick. He saw cells stop dividing after about 50 divisions. He thought, at the time, that this was just the cells shutting down. Quiet. Inert. He was wrong about the quiet part. A cell that has entered senescence is not dead. It is not asleep. It is alert in a peculiar way. It cannot divide because its telomeres are too short, or its DNA has been damaged, or it has been exposed to stress that triggered the senescence response. But its metabolism is on. Its transcription machinery is on, and it has switched into a new program of behavior that includes pumping out a cocktail of signaling molecules. Inflammatory cytokines, proteases, growth factors, reactive oxygen species. This cocktail has a name. The senescence-associated secretory phenotype, SASP for short. Judith Campisi, at the Buck Institute in California, spent decades working out what these cells were doing and why. The original idea, and it is still the best idea anyone has, is that senescence evolved as an anti-cancer mechanism. If a cell has damaged DNA, the safest thing to do with that cell is take it out of the dividing pool. Senescence does this. It is a brake. Without it, every sunburn and every X-ray and every chemical insult would have a chance of turning into a tumor. But the cells linger. The immune system is supposed to come along and clear them, and when you are young, it does. As you get older, immune surveillance weakens, and the senescent cells start to pile up. They sit in your fat, in your liver, in your skin, in the lining of your blood vessels. By the time you are 80, certain tissues can have up to 20% of their cells in this state, each one pumping out cytokines, each one calling for inflammation in the tissue immediately around it. This is what people now call inflammaging, a low-grade, steady, smoldering inflammation that does not come from any infection, has no fever, no swelling you can see. Just a chronic background hum of immune signaling that wears on the surrounding tissue, recruits more inflammation, accelerates atherosclerosis, drives insulin resistance, and very probably contributes to the cognitive decline of late life. Think of the person at 68 who feels tired in a way that has no name. Not sleepy, not sad, just heavy. Slower in the morning. Aches in the joints that come and go without injury. The mind a half step behind where it used to be. There is no diagnosis. There is no test that comes back red. There is only a low, steady wear that the doctor calls aging because there is nothing more specific to call it. That heaviness is not invented. It is, in part, a chemical state produced by tens of millions of cells in that person's tissue, leaking inflammatory molecules into the surrounding fluid every minute of every day. In the mid-2010s, James Kirkland and his team at the Mayo Clinic in Minnesota did the experiment that mattered. They took old mice. They gave them a combination of two compounds. One was dasatinib, normally used in leukemia. The other was quercetin, a flavonoid found in onions, apples, and capers. The combination, taken together for a short period, selectively killed the senescent cells while leaving healthy cells alone. They called the class of drugs senolytics. The mice that received the treatment lived longer. They moved faster. Their fur looked better. Their grip strength was higher. Their hearts pumped better. Their lung function improved. They acted younger. The same compounds are now in human clinical trials for several diseases: pulmonary fibrosis, diabetic kidney disease, frailty, early Alzheimer's. The first results are promising. None are conclusive yet. The science is moving quickly, and we will know more in a few years. The senescent cell is loud and inflammatory and expensive to maintain. The expense is the next clock. Where is the cell getting the energy to run its SASP machinery for years on end when it cannot even divide? It is getting it from its mitochondria. And the mitochondria are the fourth clock. Think about a single mitochondrion, a football-shaped body about a micrometer long, sitting in the cytoplasm of one of your cells. Its job is to take electrons from food, pass them down a chain of proteins, extract a little energy at each step, use that energy to pump protons across a membrane, and then let the protons flow back through a molecular turbine that turns the energy into the molecule that powers every other reaction in your body. The molecule is called ATP, adenosine triphosphate. 3 phosphate groups bolted onto an adenosine, with the bonds between them storing the energy. This whole process needs a coenzyme. The coenzyme carries the electrons from the food to the chain. It is called NAD, nicotinamide adenine dinucleotide. It exists in 2 forms. One is empty and ready to receive electrons. The other is loaded with electrons and on its way to the chain. The amount of the empty form, NAD+, that your cells carry determines how fast they can extract energy from food and how well they can run all the downstream reactions that depend on it. When you are 20, your cells contain a great deal of NAD+. When you are 80, they contain about half as much. The decline starts in your 30s and continues steadily. Where does it go? Some of it gets consumed by enzymes called PARPs that fix DNA damage. The more damage you accumulate, the more PARPs run, and the more NAD+ they burn. Some of it gets consumed by enzymes called CD38, whose expression rises with chronic inflammation. The more inflammaging you carry, the more CD38 you make, and the more NAD+ it burns. And some of it is consumed by enzymes called sirtuins. The sirtuins are the part of this story that connects everything I have just told you. Sirtuins are a family of 7 enzymes in the cell. They remove acetyl groups from proteins. They also remove other small marks. By doing this, they regulate, among many other things, the methylation pattern on the DNA, the response to DNA damage in the chromosomes, the maintenance of mitochondrial function, and the regulation of cellular senescence itself. Every clock I have just described to you runs, in part, on sirtuin activity. And sirtuin activity runs on NAD+. David Sinclair at Harvard Medical School has built much of his career on this insight. He and his colleague Eric Verdin at the Buck Institute and Charles Brenner, who first identified the pathway by which a vitamin called nicotinamide riboside is converted to NAD+, have worked out a great deal of the biochemistry. If you give old mice a precursor to NAD+, a molecule called nicotinamide mononucleotide, or its cousin nicotinamide riboside, the cells take it up and rebuild their NAD+ pool. The sirtuins start working again. Mitochondrial function recovers. The mice run further on a treadmill. They live a bit longer. How much longer the mice live depends on how the study is run, but the consensus number is something like 10 to 15% in a typical strain under good conditions. That is a great deal in mouse years. In humans, no one has shown a lifespan extension yet. The trials have shown improvements in some markers: vascular function, insulin sensitivity, inflammation, muscle strength in older adults. They have not shown that anyone will die any later. That work takes decades. So you have 4 clocks: telomere length, denamethylation, senescent cell burden, NAD plus concentration. Each one is being measured in a laboratory somewhere right now on a sample from somebody's blood. Each one predicts something about how that person is going to age, but not all of them predict equally well. Before I tell you the next part, which is the part where someone is going to try to sell you something, I have to tell you which of these 4 clocks the evidence actually supports, and which it does not. I will grade them, not on a curve, on the absolute strength of the evidence as it stands. The methylation clock is the strongest. Horvath's original paper has been replicated dozens of times in dozens of populations across many tissues. The clocks have been refined. Grimmage predicts all-cause mortality in the next 5 to 10 years better than smoking status, better than blood pressure, better than fasting glucose. Multiple independent labs have shown that the clock moves in response to intervention. This is solid science. If you wanted to measure how fast you are actually aging, this is the measurement to get. Call it the top tier. The senescent cell story is almost as strong in mice, and 1 tier down in humans. The mouse data is unambiguous. Clear out the senescent cells, the mice live longer, move better, look younger, and many specific age-related diseases improve. The human trials are early. Safety looks good. Efficacy on hard endpoints, like death, is not yet established. The mechanism is well worked out. The clinical translation is plausible. Call this the 2nd tier, with the senescent cell story moving up toward the 1st, as the trials report out. The telomere clock is real, but the link between any one person's telomere length and that person's lifespan is weaker than the popular press makes it sound. Average telomere length predicts something about mortality at the population level. It predicts much less at the level of any individual, because the variation between cells in one body is so wide, and because what the leukocyte average tells you about the stem cells in your gut is limited. If your telomere score comes back short, that is a piece of information. It is not a sentence. 2nd tier, with a wider error bar than the senescent cell story. The NAD+ story in humans is the weakest of the 4. Not because the biochemistry is wrong, the biochemistry is well understood, but because no one has yet shown, in a properly controlled human trial, that boosting NAD+ extends life. The supplements you can buy that claim to do this are sold on the strength of the mouse data and the early human biomarker data. Both are encouraging. Neither is a lifespan trial. 3rd tier, plausible, not yet proven. That is the honest grading. The science is moving. In 10 years, the tiers may shift. But that is where the evidence stands today. Now. Here is where it gets uncomfortable. Each of these 4 mechanisms, methylation, senescence, telomeres, NAD+, has a pharmaceutical or supplement industry built around it. And each of those industries is selling you something that, in most cases, is a more concentrated and more expensive version of what your own body would do for free if you let it. Consider the senescent cell. The drug combination Kirkland used is being developed as a clinical senolytic therapy, and the eventual approved version could cost thousands of dollars per course. The mechanism it works on, clearing damaged cells from the tissue, is the same mechanism that vigorous exercise activates. When you exercise hard enough to feel it the next day, you trigger a small amount of muscle damage. The damaged cells are flagged. The immune system clears them. Your tissue rebuilds. The rebuilding produces a slightly younger, slightly tougher version of the original. This is what muscle soreness is. This is the cellular bookkeeping being updated. The methylation clock is the same story in a different language. A study by Kara Fitzgerald and her colleagues, published in the journal Aging in 2021, showed that an 8-week intervention of diet, sleep, exercise, and stress reduction shifted Grimmage scores by about 3 years in middle-aged men. 3 years off the clock in 8 weeks, with no drug. The diet was not exotic. Vegetables, beans, eggs, some liver. The exercise was moderate. The sleep was 7 to 8 hours. The stress management was breathing exercises and meditation. Telomere length. Tim Spector at King's College in London published a paper in The Lancet some years back, showing that people in the highest tier of physical activity had telomeres that were, on average, around 200 base pairs longer than the people in the lowest tier. That is the equivalent of being 4 or 5 years younger, biologically, just from moving regularly. No pill on the market does this as reliably. NAD+. The supplement industry sells nicotinamide mononucleotide and nicotinamide riboside at prices that can run $100 a month or more. Fasting raises NAD+. So does intense exercise. So does cold exposure. So does, by a smaller amount, simply eating less of the food that consumes NAD+ through inflammatory pathways. Sugar, refined oils, alcohol. And then there are the actual longevity drugs. Rapamycin, originally used to prevent organ rejection. It inhibits a protein complex called mTOR, which sits at the center of growth and aging signaling. Mice on intermittent rapamycin live longer. Some clinicians prescribe it off-label. The dose has to be carefully managed because mTOR also runs your immune system. Metformin, the diabetes drug, is in a large clinical trial called TAME, led by Nir Barzilai at Albert Einstein College of Medicine, designed to see whether it slows the onset of age-related disease in non-diabetic older adults. Both of these drugs target pathways that fasting and exercise also touch. I want to be careful here. I am not telling you that supplements do nothing. Some of them, taken under medical supervision, in the right person, at the right dose, have measurable effects. The mouse data on senolytics is real. The human data on NMN is real, even if early. I am also not telling you that diet and exercise are easy. They are not. For many people, walking up the stairs is harder than swallowing a pill. I am telling you something else. The mechanisms exist. The body has them already. The molecules being sold to you are concentrated, expensive, single-channel interventions targeting pathways that your own body operates every day, for free, in a coordinated and integrated way, when you give it the inputs it needs. The inputs are not mysterious. Movement, sleep, plants, sunlight, connection, stress that ends. When your grandmother told you to go to bed at a reasonable hour, eat your vegetables, get some exercise, and stop worrying so much, she was not nagging you. She was issuing instructions for keeping your telomeres long, your methylation pattern young, your senescent cells few, and your NAD+ pool full. She did not know about any of these things. She did not need to. She knew what worked. The science has spent the last 40 years catching up to a Sunday afternoon piece of advice she gave you when you were 12. So pinch the back of your hand again. Pull the skin up. Hold it for 5 seconds. Let go. Watch it settle. That recoil is one signal among many. The clocks I have described are the deeper ones. They are ticking inside every cell in your body right now. Some of them you can read with a blood test. Some of them you can influence with a meal. All of them respond to how you have lived this week, this month, this year. None of them is sealed off from you. None of them is fixed at birth. None of them is running on rails you cannot touch. Move. Sleep. Eat something green today. Sit in the sun for a few minutes. Talk to someone you love. The bookkeeping is being written, right now, in the cells you will carry for the rest of your life. Tell me one piece of advice from someone older than you, a parent, a grandparent, an old family doctor, that you suspect has been keeping your clocks a little younger than they should be. I want to hear it.